Welcome, advanced biology students. This is chapter 5, dynamic activities of the cell. We're going to do part of chapter 5, and then the other part we will do following. This first part is going to focus on membrane structure and transport across the membrane, and then we'll test on that, and then we'll have another test on enzymes and energy. So ultimately, the plasma membrane structure and function. When we look at the plasma membrane, it is considered a phospholipid bilayer. It is fluid in that it does flow. Things are not static. It's not stationary, so the membrane itself moves around the cell. A phospholipid molecule has a polar head that is hydrophilic and nonpolar tails that are hydrophobic. We would consider this amphipathic in that it has both hydrophilic and hydrophobic characteristics. In describing the cell membrane itself, we say it is a fluid mosaic model. Remember that fluids are anything that flows, any substance that has the ability to flow. So like I said before, the phospholipid does not remain stationary. It does flow around the cell. Mosaic art is when you have a bunch of different pieces that will come together to form or create a picture. So when we think of the cell membrane itself being a mosaic of things, we have the major component of the cell membrane, which is the phospholipids, or which are the phospholipids. And you also have these proteins that are going to be embedded in the membrane. They also have proteins that are on the peripheral end of the membrane. Cholesterol will be embedded in the membrane. And you also have carbohydrate chains, whether attached to fats or if they're attached to proteins. We would call those glycolipids or glycoproteins. So overall, here's a generalized structure. In class, you have or will create the cell membrane. If you have it, it's a good idea to pull that out now and look at it, just to get a general overview of what the cell membrane looks like. So as far as the proteins that are embedded within the plasma membrane, the proteins are going to have a specific function for the membrane itself. The proteins may serve as channel proteins, carrier proteins. They can be proteins that are involved in cell recognition. For example, cell-to-cell recognition would be if you were to receive a blood transfusion, is it the correct type of blood? Or if you were to have a foreign body come into the cell would recognize that and it would try to destroy that particular cell. So that's cell recognition. You have to see these receptor proteins. Receptor proteins are going to allow things to come into the cell. You have enzymatic proteins, and enzymes will be our next focus. But you know, basic that an enzyme is something that will serve as a catalyst in a biological system. And catalysts are anything or any substance that will speed up a chemical reaction. And you also have junction proteins, and junction proteins are going to allow things to adhere to each other or be able to allow for passage of materials between cells. So here you can see different proteins, what the channel protein will look like, a carrier protein, the cell recognition protein, receptor protein, enzymatic and junction proteins. In general, just have a basic understanding of the various types of proteins, but as far as specifics at this time, you really don't need to know that for this particular class. However, should you take an advanced placement biology course down the road, these proteins would have a lot more significance, and you will study them in greater detail. So the passage of molecules into and out of the cell. Ultimately, we say that the cell membrane itself has selective permeability. Anything that is permeable is going to allow substances to pass through it, but because of the characteristics of the cell membrane, mainly being hydrophobic in its interior with those tails, with the other phospholipids, we get this selective permeability. So some molecules can readily pass across the membrane, other molecules cannot, and they're going to need help going across the membrane. So that's why we say it has selective permeability. So there are two types of transport. The two types of transport include passive transport and active transport.
Can I have your attention, please? Will Shane Quick please report to the main office? Shane Quick to the main office.
In passive transport, energy is not required in the form of ATP because you have molecules that are moving down or with the concentration gradient. So these are things that you're going to hear. When you talk about moving with the concentration gradient or down the concentration gradient, molecules are going to be moving from areas of high concentration into areas of where they are at a lower concentration. So when we think of concentration, the concentration is the amount of that substance in a given space. The gradient is the space to which it's going to travel. So ultimately you have this concentration gradient. And in passive transport, energy is not required because you're going from high to low. And this is passive transport. And one type of passive transport is diffusion. Simple diffusion is when you have molecules that are going to move from a high concentration to a low concentration until it is distributed equally on both sides of the membrane. At that point, we would say that the substance has reached equilibrium. Very few molecules can simply diffuse through the membrane, the hydrophobic portion of the membrane itself. So one example would be if you were to put a red dye in, the red dye would travel through the membrane until it is equally distributed on both sides of the membrane and equilibrium has been established. Remember that all substances do have some sort of kinetic energy involved or motion to the particles, if you think of the kinetic molecular theory, and it's going to be the motion of those particles that are going to allow them to move from areas of high concentration into areas of low concentration. We see simple diffusion with both gases, and we could see it with liquids. So here you're seeing it with red dye, but we also do have examples of simple diffusion with gases. One of the best examples of that is if you think of the lungs, you have CO2 and oxygen gas that are going to diffuse across the capillaries into the red blood cells. So you have the gas exchange taking place between the alveoli of the lungs and going into the red blood cells. So ultimately, those gases are going to travel from areas of high concentration to areas of low concentration. So that is simple diffusion. Another type of diffusion is facilitated diffusion. Facilitated diffusion requires a transporter, like a channel protein or a carrier protein. However, it does not require energy. So it is still a type of passive transport. So molecules are still going to go with the concentration gradient, and therefore they're moving from areas of highly concentration into an area where they are more lowly concentrated. However, these molecules might be bigger molecules, so they can't just readily pass across the membrane because they're either large or because of their polar characteristics. Therefore, they need a carrier protein or a channel protein that is going to facilitate their movement.
Please, for Ryan McDonald and
they're going to facilitate their movement across the membrane.
Ryan McDonald and Josh Westcote. Thank you.
Another type of passive transport is osmosis. Osmosis is the diffusion of water across the plasma membrane due to concentration differences. Which way the water moves is dependent on the solute versus the water concentration on both sides of the membrane. There are three osmotic conditions: isotonic solutions, hypotonic solutions, and hypertonic solutions. In an isotonic solution, water is going to be at equal concentrations on both sides of the membrane. Therefore, a cell is going to neither gain or lose water. However, in a hypotonic solution, the water concentration is going to be greater outside the cell than inside the cell. And ultimately, what's going to happen is the cell is going to take on water or gain water. In an animal cell, we call this lysis or hemolysis. In a plant cell, we call it turgid because the central vacuole is going to fill up with water and exert turgor pressure on the rest of the cell. The final osmotic condition is a hypertonic solution. In a hypertonic solution, the cell is going to lose water. And it loses water because water is at a greater concentration in the cell than what it is outside the cell. So water is going to move out of the cell into the extracellular environment. In a plant cell, we call this plasmolysis. In an animal cell, we would call this state being shriveled. So the cell would shrivel up. And here you could see those examples of what would happen in both the plant cell and the animal cell. So here you could see plasmolysis. Here you could see how the plant cell is very turgid. And then here you could see a plant cell that is in a normal or flaccid state. Here's a normal state for the red blood cell in an isotonic solution. In a hypotonic solution, the plant cell would get so big it could possibly burst. Hemolysis, or the cell will become lysed. That happens because the animal cell only has a cell membrane, but a plant cell could become very turgid and not burst because it has that additional cell wall, which is a very rigid structure. And then here we have the hypertonic solution where this will be plasmolyzed, or the cell here will shrink or shrivel. Active transport and bulk transport are going to require energy input. In active transport, molecules or ions are going to move across the plasma membrane, accumulating on one side of the cell. And basically, the movement of the molecules is going to be against their concentration gradient. So in this case, it requires a carrier protein and an input of energy. Now remember, when you're moving against the concentration gradient, this is unlike passive transport in that the molecules are going to go from an area where they are lowly concentrated and being pushed or pumped into an area where they are more highly concentrated. In passive transport, it was the reverse. The best example of this is the sodium-potassium pump. The sodium-potassium pump undergoes a change in shape when it combines with ATP, allowing it to combine alternately with sodium ions and potassium ions. So if you look here, you can see the carrier protein itself, and you're going to push molecules inside the cell where they're already highly concentrated. So if you look, you have three particles outside the cell here. There are many more inside the cell. So that substance is going to come into the carrier protein. The carrier protein is going to gain energy, or we say it will be phosphorylated. And here you can see that here in the second picture. And when it becomes phosphorylated, it's going to change its conformational shape. So the shape will change and therefore open up to the inside of the cell and release that substance inside the cytoplasm of the cell. And then the phosphate group will remove itself from that carrier protein. We'll look at the sodium-potassium pump in greater detail. The other type of active transport is or bulk transport. Bulk transport is when you're going to move fluids or particles. They can be either brought into the cell or moved out of the cell. This process would be known as endocytosis or exocytosis. Endocytosis is when molecules or substances are being brought into the cell. Exocytosis is when molecules or substances are being pushed out of the cell. Basically, in endocytosis, it's being brought into the cell by a vacuole formation, or in exocytosis, you're going to have a vacuole or vesicle push up against the cell wall, and it's going to be out of the cell by evagination. The best two examples of endocytosis would include phagocytosis, pinocytosis, and receptor-mediated endocytosis. In phagocytosis, that occurs when a large material is being brought into the cell. We consider phagocytosis a form of cellular eating. In pinocytosis, it occurs when vesicles form around a liquid or very small particles, and they're going to be brought into the cell. Therefore, we commonly refer to this as cellular drinking. And then, of course, there's receptor-mediated endocytosis. And ultimately, what's going to happen there is receptors for particular substances that are found on the cell membrane are going to recognize those substances, and they're going to be able to carry out some sort of response to bring in a particular substance or molecule. And we'll look at that in class 2. So here you can see an example of endocytosis, where something is being brought into the cell. And up here would be exocytosis, where something is being pushed out of the cell. Malfunctioning plasma membranes. There are some diseases. Diabetes type 2 is when you have failure for insulin to bind to receptor proteins, but the number of carriers sent to the plasma membrane is not enough. So insulin does bind and yet don't have enough carriers, and ultimately what happens is you get too much glucose in the blood that results in a spillover into urine, therefore increasing your glucose levels in the urine. Color blindness is usually 3 types of photopigment proteins in the plasma membrane that have photoreceptor cells for light. Some people lack functional red or green photopigment, therefore you would have color blindness. And there's also the disease of cystic fibrosis. And cystic fibrosis is when you have unregulated passage of chloride ions through a plasma membrane protein channel. Ultimately, what happens then is you get a thick mucus that is going to appear in the lungs and the pancreas, and it's going to damage the lungs and ultimately contribute to an early death. So here you can see an example. This kid is undergoing percussion treatment. That helps loosen up some of the mucus in the lungs for it to be expelled. But you could see overall on how you have a defect in the CF gene on chromosome 7, which ultimately is in the DNA, and it transcribes for that protein that doesn't is defective in getting rid of chloride ions. And that deals a little bit more with genetics and molecular biology, which we'll get to later in the year. Cell membrane communication. The cell membrane does communicate with one another. We looked at some of these already. Extracellular materials allow cells to join together and communicate. Plant cells, all plant cells have primary cell walls. Living plants are connected by those channels called plasmodesmata. In the animal cell, you have those anchoring junctions, which in tissues are going to allow it to stretch. You have tight junctions, which are zipper-like or are going to fasten close to not allow any leakage. Gap junctions are going to be used for strength and coordination. And you also have that extracellular matrix. And it varies from very flexible to very rock solid. And that ultimately, the extracellular matrix, its composition includes fibers and glycoproteins. And there you could see examples of animal cells and their extracellular matrix. The evolution of the plasma membrane. The plasma membrane must have arisen early in the evolution of life, similar in bacteria and eukaryotic cells. It's going to serve as a barrier while still allowing transport to take place. Phospholipid component is going to form that boundary between the internal cell cytoplasm and the extracellular parts of the cell or environment. The evolution of multicellular organisms would have been impossible without extracellular material. And that's it for this chapter. Thank you very much for watching and have a nice day.